Child. All right, I'll just take a few quick minutes to introduce Dr. Young here. Dr. Young is currently our PGY6 chief resident in the Combined Neurology Psychiatry Training Program. And to give a little background on Dr. Young, he's kind of a Worcesterite. He attended Clark University, where he earned degrees in biology and environmental science and policy. After completing his studies at Clark, he promptly moved out of Worcester and had no plans of returning. Just kidding. He subsequently attended medical school right here at UMass in Worcester. Prior to attending medical school, he worked in the field of genomics at a biotech startup called Genomics Collaborative. And in addition, worked at the Broad Institute at MIT and Harvard, where he contributed to large-scale genomic research projects. You'll see how this has shaped his presentation today and his interest in these topics over the past decade. So as mentioned, he has interest in genomics, connectomics, which is kind of the way that we neuropsychiatrists look at the brain and how things are interconnected, and how that relates to things like cognitive disorders, schizophrenia, and natural language processing. I will allow him to explain that to you if you are interested. I will add, just from a personal note, Dr. Young is a marker in our program for me, as he started his residency intern year the same year I graduated and started working in the training office. For any of you that already know Dr. Young, and I'm imagining that's many of you, because he has made his mark year for the past 10 years, by the way, if including medical school and his training. He's easily recognizable as an extremely affable, friendly physician with a gentle and caring nature at the bedside. During his epoch here at UMass, he's made wonderful friends and colleagues, and also started an extremely adorable family, and I'm sure he would share photos with you if you ask. What his approachable countenance belies is an intimidating intellect that can make very easy work of very hard things. I expect we will see a representation of that today, and I personally cannot wait to see what's next for Dr. Young. All right, so let's all welcome him today for his grand rounds.
Thank you. Thank you. I'm blushing. Is it too late to change my topic? Okay. So we're going to start I'm going to talk about non-coding RNA and neuropsychiatric disease. And this is something that's kind of been percolating in the back of my mind for quite some time. And I've been doing some research on it. And hopefully we all learn a little bit and in a non-intimidating manner. Disclosures, I don't have any conflicts of interest in relationship to this program. Brief outline, we're going to define long non-coding RNA. We're going to discuss the role of these IncRNAs in regulating gene expression, talk about a little bit of my research related to IncRNAs, and then we're going to try to dive in a little bit into how IncRNA might be involved in neuropsychiatric disease. This is a very broad field, so I'm going to try to kind of keep it focused. And we're going to start out with a reminder that RNA is central to life. This is what's known as the central dogma of molecular biology. You've all probably had this embedded in your brain. DNA makes RNA, makes proteins. The protein carries the code and from DNA, and it's translated into proteins. And so this is particularly fascinating to me because this is a system of codes that's interpretable and it serves a purpose, and it developed spontaneously. Which to me is kind of magical. But and when it comes to humans, there was a time where we thought we were like the most impressive things in the world, and maybe we still are. But before this thing of the Human Genome Project, it was estimated that the humans had like over 100,000 genes. We said, we're these incredibly complex individuals. Our brains are so much bigger. We can think, we can do all these things that other animals can't. And then it turns out that we only have about 22,000 protein coding genes, and that's about the same as everyone else. In terms of primates, we're 98.8% genetically similar. Cats were 90% similar. Dogs were 94%. Even the fruit fly, we share like 60% of the genome. And so our initial estimates of there's 100,000 genes came down to about 22,000 protein coding genes. And then there's about 34,000 transcripts, or this is RNA that's transcribed from DNA that was initially termed junk DNA. And then slowly, the field has started to realize that maybe it's not just junk DNA. It may be all of this extra RNA that our genome is producing and our cells are producing are having a function. So this, I just borrowed this from the New York Times because it's a nice little picture. But potential roles where there's genetic switches, there's regulation of gene expression, and then maybe that all of this extra quote unquote junk DNA contributed to disease in some way. A brief summary, if you look at this pie chart here, there's all of this junk DNA. It falls into categories: non-coding RNAs, ribosomal RNAs, transfer RNAs, transposable elements, introns, intragenic regions. We're really concerned today with the non-coding RNAs. And then when it comes down to non-coding RNAs, there's several different types. Somewhere along the lines, they were divided into small, meaning less than 200 nucleotides, and large, meaning greater than 200 nucleotides. I don't know if there's a biological reason for that. It seems somewhat arbitrary. And I was almost able to track down the initial reference of when someone finally first described a long non-coding RNA. But once you get into this category of long non-coding RNAs, there are, again, more subtypes. Intergenic, here you have between gene A and gene B, there's a stretch of DNA that codes for they call it lincRNA, which is the same, but long intergenic RNA. It can be within the intron of the gene. So you have a gene, you have several exons, the intron is spliced out, the exons are put together. So it can be within that intron. It can be an antisense RNA, meaning it's the same region of the gene, except you're transcribing it in the backwards direction. And then there's enhancers and there's a few other types. But these are kind of the big ones that we think about: the intergenic, the intronic, and the antisense. Okay. This is a scary slide. I'm sorry. I'm not going to go through this whole slide, but the moral of the story is that lincRNAs, it regulates gene expression on many levels. So the important thing, it's actually probably a little bit hard to make out. The important thing is here, we go from A to R. So most of the alphabet are different ways that lincRNA can regulate gene expression. And it spans a lot of the molecular functions. So modifying the chromatin state, the epigenetic modifications, recruiting transcription factors, alternate splicing. So using the same gene and making different forms of a protein, and then modifying the RNA and protein after the fact. Also, messenger RNA stability and decay. So if you have a messenger RNA, if you want to make a lot of that gene, you kind of keep it around, and if you don't, you get rid of it. And then this is another scary slide where I don't mean to scare you, but this is kind of busy. But eventually, the field has figured out that some of these lincRNAs actually do produce a protein. So you have this, your little lincRNA, this is your open reading frame, and it creates a small polypeptide, and this apparently has some function. This is kind of a graph of the function of the micropeptides produced by lincRNAs in gastrointestinal cancers. But really the only point is some of them actually do produce peptides and proteins. Okay, And now the interesting thing about these is, maybe they do make us special. So this is your kind of evolutionary time scale. And as you see over time, we go from single-celled organisms, primates, and the amount of non-coding DNA increases with organismal complexity. And then more recently, we have studies that show that up to 40% of known lincRNAs are expressed in the brain. Thousands of them emerge with primates and humans, so we don't see them in any other species. And then hundreds of them are specific to humans. That's pretty curious. And then this other fact is some of them are conserved and some of them are not conserved. And when we think of like a conserved gene, it's a gene that looks similar in a mouse and it does the same thing in a mouse, in a primate, and a human. So many of these lincRNAs, they don't have a functional homolog in a lower species. So they make it pretty curious. And then there's a lot of evidence that they facilitate neuronal development. So across this time, you have your pluripotent cells. Some of these words are really kind of, phrases are funny. Neurogenic commitment, cell fate choice. Like, this is what I'm going to be. And along the way, the lincRNA help make this decision and help push your undifferentiated cell into a neuron. And then once it's decided to be a neuron, it helps it be a better neuron and regulate the synapse formation. So there's all of these ways that, okay, maybe this is interesting. All right. And then there's even more evidence that it mediates synaptic plasticity. And so here in this graph, this lightning bolt, I guess, is meaning that it's getting some signal from another neuron. It's not actually being shocked, or maybe it is, I don't know. And so the lincRNA can facilitate activity-dependent gene expression. And that can do it many different ways: alternative splicing, expressing the genes, and then down at the synapse, it can regulate the growth of the axon and the synaptic density. So these are pretty interesting features of the genome that may be important to us as psychiatrists and neurologists and neuropsychiatrists. All right. But most of these lincRNA remain uncharacterized. And there's been a lot of efforts to explore this computationally, and I tried as well. So we're going to talk a little bit about that. So quick overview. What I did was I created a composite network of gene expression data and protein-protein interaction data. And I'm going to talk about this quickly. We optimize the density of that gene expression network, and then we propagate information through the network to help us figure out what this lincRNA might be doing. And then we evaluate, does this make sense or does this not make sense? And then the data sources I used was the Genotype Tissue Expression Project, abbreviated GTEX. There's a protein-protein interaction data from the Human Integrated Protein-Protein Interaction Reference, HIPPIE. There's functional annotations from the Gene Ontology. These are called like gene sets. It's a collection of genes that do a certain function. And then I have a network propagation algorithm. So talk a little bit about the gene expression data. So this is a project where they collect post-mortem samples from a rapid autopsy. And the idea is, let's collect as many tissues as possible. There's even samples from 13 regions of the brain. And they quantify the gene expression by basically counting RNA in each tissue sample. And I didn't use this data, but they also collect genotype data. So the idea is you have variation in your genotype data, and perhaps that explains the gene expression data. I'm really just using the gene expression data. And this was a total of 544 donors. This was a little bit of an older data set, but there's 8,555 tissue samples. 193 of those donors also had brain samples, and it had about 1,250 brain samples. And then kind of more defunct data, I was looking defunct. Additional data, I had about 27,000 genes, 18,000 of those were protein coding, and about 3,600 were lincRNA. So that's kind of the initial gene expression data. And once you take that data, we create a gene co-expression network. And that sounds like a fancy term, but all we are really correlating the expression levels of the genes. So I have a little table here. We have gene 1, gene 2. Going across, you can see they correlate a little bit. In gene 2 and gene 3, they look like they are inversely correlated. We take the absolute value of the Pearson correlation coefficient, and our goal is this is a little model of our gene co-expression network. Now, we initially calculate all pairwise correlations, which is like, what is it, 733 billion, which is far too many. So we want to trim that down, and we'll talk about that in a little bit. This is our goal here. So we have our genes, and these are how they are connected to each other by their absolute value of their Pearson correlation coefficient. And then the individuals who created this HIPPIE resource have chosen a hippo as their logo. I'm not sure why, but it's stuck and it's pretty cute. But this is, protein-protein interaction. So from the available literature, they kind of comb through all the resources and they say, okay, these genes are connected, are interacting with each other. They have a confidence score of how well they're connected or how confident they are in the interaction. I had about 15,000 protein coding genes, and there was about 260,000 interactions. And then so this is the same genes, but let's say for the sake of example, we know that these orange lines are the connections between those genes. So we have our gene expression data, and then we have a known protein-protein interaction data. And then we kind of combine the two networks. This is the combined network. I've highlighted, where the connections are shared in yellow. And I'm not going to go through a lot of this like naive Bayes stuff. Basically, that means we assume they're independent, even though we know they're related, but it's too hard to model. And so when the gene expression and the protein-protein interaction data agree, that emphasizes the connection between these two genes. And I mentioned these functional annotations over here on the right. These are what would be called gene sets. So I have all these 10 genes. These are just the names of the genes. And they all participate in this function related to the juxtaparanoid region of the axon, which is nice. We like that. It's related to the brain. Obviously related to the brain, makes us feel good. And they all have an ID. They fall into 3 categories of molecular function, cellular component, biological process. And these are readily available online. So I start with about 3,360 just to train my model, which I will talk about in a minute. And then eventually I use over 7,000 to make my functional predictions about the lincRNA. And so we use something called 5-fold cross-validation in this network propagation algorithm. So I'm not going to go through the whole algorithm, but looking at this model here of our network, I have gene 1 and gene 3. They're highlighted. So we're pretending that they belong to this sample go category. It does some function, and we are using these 2 genes. So the idea is we know that these genes are related to this function. So we're going to simulate stimulating the network at these points, and the information is going to flow through the network down to our link RNA. And so it's a little bit of a guilt by association method. And let's say if we had an example where gene 2 and gene 4 were involved in a known function, we'd probably say, oh, that's really related, and then gene 1 and gene 3 are maybe not as related. For this cross-validation, we divide the genes into 2 groups, well, into 5 groups, but here I'm using 2. And then we hide the label. So we hide the fact that we know that this gene 3 is related, and we just stimulate the network here, and we see how well we can recover that label. And then we do that for the next one. We hide this label, and we stimulate the network here and see how well we recover this. And we calculate the area under the receiver operating characteristics curve. You guys probably know this from literature at AUC. And so we do that for 5 folds, and we do that for all of our terms. And then we average that all together to figure out how our model did. And so averaging overall that, the area under the curve is 0.876, which to me is good enough. You know, that's like right in the range of like, this is pretty solid. You know, I found some references that say that, you know, over 0.9 is really kind of hard to get to. But in this graph, this red line, this is just the gene expression data. And so what we're doing here down at the bottom is we're adding more genes to our network. And as you can see, when we start with very few and we add a few more, we get some more information, and that's helpful, but it kind of plateaus. So adding more genes to our network or connections to our network isn't necessarily helpful. This spot right here, this one right here, is just the protein-protein interaction network. So we get a lot of information from that. And then when we add more genes or from the gene expression data, we get a little bit of an increase, and it plateaus, and it actually almost looks like it decreases. When we start to add too many connections, we get more noise in our network, and it's subtle, but when you're thinking about computational cost, like you have computers doing all these calculations, so you want to have efficient calculations. And so this is the network that we chose right here. I think it makes sense that the protein-protein interaction network provides a lot of this information, because the gene sets are probably drawn from the same information that the protein-protein interaction is. And then, so we choose that network. We say, okay, we're going to use this network. And then to make our functional predictions, we do some permutations, and I'll go through that, and we calculate a z-score. So this is our sample, functional category, gene 1 and gene 3. And a permutation means that we just switch the label. So instead we say it's related to gene 2 and gene 4. And then we say maybe it's related to gene 5 and gene 4. And we do that 50,000 times. You can't do that with this many, and then we get a z-score, which means this is the number of standard deviations above or below the average for that gene. And then essentially we get a z-score for every functional category for every gene, which is too much. And I was like, I need to figure out how to make this more accessible to myself, because I was like, I need to understand if my results are actually any good. So I created a website. I have a few minutes. So I can do a quick demo, if I can remember how I got out of this. Escape, there we go. And here, okay. And this is the website. Anyone have a favorite gene? I use this, it's called Exist. This looks like a big gene crowd. C9orf72, right? Frontal temporal dementia, ALS. ApoE4. I like Exist. This is a X inactivation specific transcript. So this is kind of like a prototype of a link RNA. So it's responsible for the epigenetic modifications that silence that extra X chromosome in the XX cells. That has a lot of X's. But it's an epigenetic modification in gene regulation. So And then I arbitrarily set the z-score 3. That seems like 3 standard deviations above the mean is great, but I can change this if I want. And then it shows me all the things that this model has predicted. And so these z-scores are pretty high, 27, 10 through 10, and this is just the top 10. And, you know, there's some histone, demethylation, demethylase activity, regulation of depolarization. So some of this kind of makes sense of like, if you're doing an epigenetic modification, these are things you would expect. I don't know about the vocalization behavior on neural cell adhesion. Who knows? And then one of the reasons I put this together is so you could explore. You're like, I don't really understand all of this. We can look at the genes that it's connected to in the network, and then we can plot all of these functions, right? It starts with a simple plot, but, you know, I can add a lot more, and then I can add some more genes. Oop, too many. You kind of end up with these hairballs, and then I can play around. Idea being, let's group everything together so it makes sense. And it really just a way to explore what the data means. All right. So this is how I kind of evaluated whether my predictions were adequate, as I went to the literature and I said, okay, this is a lincRNA. We know something about it. And then what are my predictions? And back to the presentation. All right, and then I have to switch this. Okay. All right, so you may have noticed there's this lincRNA MALAT1. And it was in that one of those initial graphs as it regulates synaptic density. And there's a fair amount of information or publications that suggest that this regulates gene expression and also is involved in alternative splicing. There's one reference where it was called one of the 3 musketeers of the synapse, which is a phrase I never thought I would hear or read, but here we are. And there's evidence that it is involved in the rapidly modulating neuroplasticity response to stimulus processing. So maybe this is one of those things that helps your neurons, you know, in learning and responding to sensory input. And then there's evidence that if you knock down this lincRNA, it decreases synaptic density. And if you increase it, overexpress it, it increases synaptic density. And so I went looking to the, you know, literature, PubMed. And this is an older study, but it's upregulated in the cerebellum of people with alcohol use disorder. So you can see these plots over here. This is the cerebellum right here is the main one. It's also upregulated in the hippocampus and in the brainstem. I don't know where they sampled in the brainstem, but it's not upregulated in the cortex, in the frontal cortex or the motor cortex. And then they gave a lot of alcohol to rats to see if that did the same thing, and it didn't. But it did when the rats went into withdrawal. They showed expression in the cortex, not the cerebellum, and they thought that maybe this is in response to this excitatory input. When we withdraw the alcohol, genes get all this extra stimulation. Okay, so that's interesting. And then I found another study where they gave rats TBIs, and then they gave them intranasal MALAT1, and it improved the recovery in the rats. So I'm not sure what the elevated body swing test is, but I think you pick it up and swing it back and forth. and you somehow quantify what the rat is doing. But in the green, this is the TBI. This is just the TBI. And then in the purple, this is when they gave them exosomal mallet, the exosome without the mallet. And then this is when they gave them an exosome with the mallet. So, okay, there's a motor function. And then radial arm water maze, guessing you just put them in water and have them swim through a maze. And again, this is the green is the standard TBI who got no treatment. The blue are the people who are rats who got the intranasal mallet, and you can see that there's an improvement. Okay, and then continuing on with this MALAT1, there was a study in 2020 that showed that it was underexpressed in the blood of people with bipolar disorder. And when they used this as a biomarker, there was an area under the curve of about 0.8. I do want to point out that I did not see, I was, okay, well, maybe this is a biomarker. I didn't see a correlation between the gene expression in the brain and in the blood. That doesn't mean that this is not true, because my gene expression data are quote unquote normal people, or mostly normal people who have illnesses related to aging and not necessarily psychiatric illnesses. Okay. So, and then there's a lot more information on MALAT, that it's associated with many other non-neuropsychiatric diseases: tumor genesis, myocardial infarction, endothelial function, diabetes, diabetic retinopathy, some evidence that it helps in stroke, and some evidence that it may contribute to Parkinson's disease. And okay, so looking at it, it is not specific to the brain. These are all the expression values in all the tissues. This yellow cluster here, this is the brain, most expressed in the cerebellum, which is interesting, but it's also expressed in many other tissues. And then I create this nice little plot, and I have a big cluster here that's related to gene expression through microRNAs. And then if you look, there's a whole bunch of others that could also be indicated that this functions in gene expression. And then there's a cluster here that looks like it's related to splicing. Okay, good job, Taylor. Maybe that's a success, I don't know. So moving on, there's another MEG3 is maternally expressed gene 3. So this is an imprinted gene. This is a picture of the RNA. I'm not sure what the kissing loops are, but they're noted. And there's a few interesting features about this that may make it a biomarker. So it was overexpressed in the peripheral blood of children with autism disorder, and the AUC was 0.79. It was underexpressed in the peripheral blood of patients with bipolar disorder. And so this is 2 biomarkers now for bipolar disorder. And the area under the curve was 0.63, which is not as compelling, but interesting to note. It was underexpressed in the plasma of patients with Parkinson's disease. And this was actually pretty compelling, an area under the curve of 0.88. And then in a separate study, the expression level in the plasma was downregulated in Parkinson's disease, but it was inversely correlated with the non-motor symptoms. So the non-motor symptoms of Parkinson's disease are like the depression, the constipation. And then it was correlated with the score on MoCA and mini mental status exam. So the less of this they had in the blood, the worse they did on the MoCA.
Can I interrupt you?
Yeah.
So I'm a little confused. Did you say this was maternal?
Well, yes, it's an imprinted gene. So it's maternally expressed. So your maternal, the copy of your maternal chromosome is the one that's expressed. The one on the paternal chromosome is not expressed.
And for bipolar disorder, is this a state or a trait? I mean, does it depend on what phase of the illness is someone, or you just have this? It's a gene that's just always there.
I don't know if they measured it during like an acute manic or mood episode.
Well, the reason I ask is because in Parkinson's, you said it correlated with a symptom.
Yeah. So I think it was just the trait of these people have bipolar disorder.
Okay.
Yeah. Okay. And again, I looked in my relatively normal data set, and I did not see a correlation between the expression levels in the brain and in the blood. That does not mean that this has not been observed in the literature. So the function of the brain isn't well defined. I spent some time trying to figure out if it's conserved. Partially conserved might be the answer, but then I found that it was highly expressed in the developing and adult mouse brain, and then deletion leads to perinatal death. In stem cells, when they took them into a group of cells that underexpressed it and ones that expressed it higher, it showed a reduced neural differentiation potential. And then there's evidence that it modulates expression of the AMPA receptor during long-term potentiation, and then it may mediate CNS injuries. And so this table here is admittedly a cherry-picked list of my predictions, but I'm showing you the rank here. So this is where it falls in that whole list, and this is the Z-score. Now, regulation by expression of genetic imprinting, we'd expect that because this is an imprinted gene. And then there's a nice smattering of things that are easily identifiable as functions related to the nervous system. And way down on the list, I threw this in here because I was curious. Maybe I have some evidence that it's related to the AMPA glutamate receptor as well. But that is not as compelling as a predicted function. Okay. And then moving on, we're in the vignette phase here. So this was an interesting study I found of this IncRNA 473. It doesn't have a formal name, but it was associated with depression in women. And these were post-mortem brain samples of men and women, some of which were control, some of which were had depression. And then there were 6 brain regions that were sampled. And they found a number of genes that were differentially expressed in both. And then they narrowed it down to this one because it was expressed in neuroprogenitor cells, and it was something easy they could study in the laboratory. And it's primate specific. And then they did something interesting. They transfected into the mouse brain with viral, and they found that when they gave it to, they had the mice express it, it promoted stress resilience in the female mice only. And it promoted changes in synaptic function in the female mice only. And changes in gene expression. And this was only in the female mice. So they gave it to male and female mice, and then it promoted these changes. They did some other experiments, and they thought maybe it's related to the cyclic AMP Krebs signaling and neuronal activity. And those are things that are, may regulate the gene expression in the brain. And then there's evidence that it was upregulated in several types of cancers. And then it's got some diverse functions. So when I just dig a little deeper, as I mentioned, cancers. And then there's some evidence that it's downregulated in the placenta of women with severe preeclampsia. And there's evidence that it's required for decidualization of the endometrium, so preparing the endometrium for pregnancy. And then there was some evidence that it may be a biomarker for stroke. And then, I don't have like a great story on this one. Related to the brain, I have something that's like, this is just the top 15 predictions. And, this is representative. I kind of combed the list. I have like neuropeptide signaling pathway, apoptosis, that makes sense with cancer. I have ovarian follicle development. And then I can't even say all the hyaluron, the things that sound like joint supplements for your dog. There's a bunch of those. And yeah, I wish I had a great story on this. If I have a little bit of intrigue in that when you look at where it's expressed, it's highly expressed in the ovary. And I guess that kind of makes sense based on my prediction. And also, you know, when the follicle matures, you do need some apoptosis. It's a little bit more of a curiosity, and I don't have a great story other than like, this is kind of interesting. Okay. Hairballs. These are called hairballs. This was a study where they looked at gene co-expression networks in brain samples. There are two data sets. This is someone else's data, and this is someone else's research. There was a GVEX data set, which are control adult brain samples, and then there's a developmental brain sample data set. And they identified this lincRNA called DGCR5, which is stands for the DeGeorge syndrome critical region 5. So this is within the 22q11.2 duplication area that you see on every psych board exam. And these were independent gene co-expression networks. We call them hairball because they're showing that just every gene is mostly connected to every other gene. And in the center, you have this DGCR5 and then DGCR5. And then the color codes are genes related to schizophrenia or differentially expressed genes. So the red are genes identified by the genome-wide association studies. Yellow are genes that have de novo mutations. And then there's the differentially expressed genes in either. So there's a few interesting things about this. I mentioned it's in the 22q11.2. It is primarily expressed in the brain, more so in the cerebellum than the cortex. And then there's over 1,000 what are called EQTLs, expression quantitative trait loci. And this is a picture meaning that you have a genotype of TT, TC, or CC, or whatever the genotype is, and that affects the expression level. So here you can see that people with are heterozygous for the C, they express lower than people who are heterozygous for the T. And then here's just another example where the G, heterozygous for the G is less expression than the A. And these are samples in the frontal cortex. And this is stuff that you can just go to the GTEX portal and look this up. And then this is my top 15 predicted functions for this one. Optic nerve development, neuroretinal development, glutaminase activity, gamma catabolic process, GABA A receptor, dendritic spine development. So these make sense. This is a good story. I like this. Okay, so good story. And I think I have like one or two more vignettes and we'll wrap up. This was a very interesting study. So these were postmortem brain samples from individuals with autism. And this group of researchers identified 60 lincRNA that were differentially expressed in their brains. You can see that these are the red, these are the people with autism. And then if you squint and look down here, you can see how these are blue and these are red. And that really means those are the differentially expressed genes. And many of these, they reported had a primate-specific expression pattern in the brain. And I had data on 57 of the 60 genes. And this is the region. So the gray bars, is either the frontal cortex or the temporal cortex. Age is kind of hard to make sense of. So I make my predictions. And what I did not show you on this website is that you can put in more than one gene. You can just put in a whole list of genes. I put in a whole list of genes. And I get a summary of the functions. So I had 16 genes that were predicted to function in this chemical synaptic transmission. And then you can see there's a whole lot that looks like it's related to the nervous system, and that many of these link RNAs may be participating in the same function related to the development of the nervous system. The cilium looks like it stands out, or the cytoskeleton, but that's really important in terms of getting those neurons to the right place and receptor signaling. And just a few more kind of notable things. The role is expanding. So this is a really interesting one, the motor neuron disease. So I mentioned the C9orf72. This is there's a repeat expansion that increases your risk of FTD and ALS. And there's actually an antisense version of it. That's a link RNA that may be involved in developing motor neuron disease. I did not have data on this particular link RNA in my data set. And then in terms of like brain injury, stroke, and hypoxia, there's starting to understand that link RNA may be helping to regulate that. And then I don't want to spend a lot of time on this slide, but you've probably maybe heard of this BDNF Val66 MET mutation. There are a lot of publications on it. Is it involved in synaptic plasticity? And there's evidence that maybe it affects your response to TMS. It turns out in the GTEX data set that it's, again, an expression quantitative trait loci for the antisense version, which is a link RNA. And here's my, the schematic I showed before of BDNF and the relationship to the antisense. Interestingly, if you look at the GTEX data, they identified 730 of these eQTLs related to the antisense BDNF link RNA in the brain, and 251 in peripheral nerves. So this mutation that we've studied a lot may be related to link RNA, but then there may be other mutations in the genome that also modulate that link RNA. So a little bit of a interesting story about how complex the genome is and how that complexity may help regulate synaptic plasticity. And then, all right, conclusions. RNA rules the brain, maybe. So there's still some debate that whether link RNA are truly functional. There are some people who think it's just transcriptional noise, that you're producing proteins and then the rest of the stuff just gets produced as a byproduct. The role of link RNA in neuropsychiatric disease has been emerging and continues to emerge. And then many of my functional predictions are plausible. Requires further investigation. I don't think you can draw any hard conclusions. But one of my goals in this was to create a resource. So when someone says, oh, I have this gene, I'm interested in it, what does it do? They can go to this website and maybe learn something and develop a new hypothesis. And the link RNA may be one of the bridges between the genome and the connectome. So when you're thinking about the genome in terms of the brain and psychiatric disease or neuropsychiatric disease, you're ultimately thinking like, how is this affecting like the connectome, right? So if you have a SNP that's identified with a disease, your hope is that you find some way that this changes the way the brain works. And that's all I have. But thank you to Dr. B and Dr. DeGrush for indulging me for 6 years. And Jiping Wang was my mentor on the genomics project.
Thank you. And do we have any questions? And we do. And we have some online also.
So Taylor, thanks for the 101. I'm interested to find out when it gets complicated. That was tremendously easy to follow, very clear. No, actually, but anyway. So the question I have is, a couple of years ago, we learned that sometime in adolescence, pruning seems to play, at least the genes involved in pruning seem to be responsible for more variants than a lot of other candidate genes in schizophrenia.
Okay.
And I'm wondering whether the amount of this non-coding lincRNA is constant over lifetime, or can you watch the amount change with certain ages so that it would make you suspicious that there's a major neuroplasticity event happening if you knew that it was changing?
That is a great question, and I don't have an easy answer for you. You know, the studies I mentioned where it's in the peripheral blood, the question is, how do you measure that? I guess in mice, but I think there are, at least in the developmental trajectory, that there's probably some time frames where, okay, the mouse is going through this developmental change. We could probably measure it at that point, but I didn't look into that specifically. But I think that's a very interesting question. Yeah.
But overall, just from seeing you demonstrate this, it looks like you could have a ball going down the rabbit hole of what, when one function is being called on to be decoded, or like, what are the other things that could be nearby and being turned on, you know, so.
Yes, and then, you know, this is like I said, this is a mostly normal data set. There are data sets from brains, developmental brains, and, you know, so there's other data sets that would be an interesting place to look.
All right, we have a question from Dr. Banerjee.
Yes.
In the process of screening for NCRNA related functions, could you utilize CRISPR to explore downstream effects of not having that particular sequence of NCRNA?
Yes. That is kind of one of the areas of their research now, is that they use CRISPR to knock down genes and to edit them. You can do that. I don't do that. I don't do much of the wet lab work. I'm more of a computer person. But
I have a non-genetic question.
Sure.
Why is TMS being given to stroke patients? Is it for depression or something else?
Motor recovery.
Motor recovery.
Yeah. So the idea being is that you've disconnected, you know, part of your motor cortex from your ability to move it, and if you stimulate those areas, maybe there's an improvement in motor recovery and functional recovery.
Is this something that's done routinely clinically? Is this still research?
It's a research area. Yeah. The neurologists are learning about TMS. They're starting to pay attention and they're getting interested. Or in, you know, Mike Fox does a lot of research with Parkinsonism, and he's shown that if you stimulate the motor cortex with TMS, it improves the motor symptoms of Parkinson's, but not the depression.
Hi, new here. I'm Ryan Logan.
Hi.
In psychiatry and neurobio, we do a lot of computational functional genomics work in both human brain and animal models.
Okay.
So the research that you created is pretty interesting. There are other long non-coding RNA resources out there. So my first question is, what makes yours different? And then the second question I have is, just playing around with it for about 10 minutes while you were talking, it actually seems pretty valuable. Do you envision integrating this with disease data sets? There are a significant amount of human postmortem RNA sequencing data sets, single cell data sets, proteomics data sets, multiomics data sets that you could potentially integrate with. And I think you touched on some of those in the vignettes.
The one thing that makes it different is this was one of the first ones, or at least from when I started the project, that used the GTEX data set and also the protein-protein interaction data set and this specific algorithm. There's tons of papers about people making resources and using gene expression data, but I think the combination of gene expression and the protein-protein interaction data was fairly unique at the time, and the specific algorithm I'm using to make the predictions. And also the website I thought was something that made it more unique in that, like, usually you just get spreadsheets, or you're like, you can download stuff. Like, this had a way to explore what actual predictions might be. I don't have any immediate plans, but as I was presenting this, I was like, oh, I know there's all these resources and, you know, it's definitely an area that would be pretty easy to just kind of swap in some new gene expression data and, you know, add on to the knowledge base, particularly with brains.
Super cool.
Thank you.
Any other questions? All right, well, we don't have any more questions online. Thank you very much. It was very interesting.
Thank you for listening.